Welcome to Catch This. My name is Trudy Ray. I'm going to try to veer away from coronavirology slowly because there are so many cool virus stories out there that people just don't know about, like this evolutionary dance between viruses and bacteria. Seriously, this is some cool stuff. Surely you know that there are viruses that only infect bacteria. They're called bacteriophages, or phages for short. And these phages could be very beneficial to us in this age of increasing antibiotic resistance. They could act as therapeutic agents alternative to antibiotics or possibly in combination with antibiotics. But did you know that phages don't just randomly infect bacteria when they're there, but they are also capable of monitoring when would be the best time for infection based on the abundance of bacteria? And they do so by eavesdropping on the cell-to-cell communication between bacteria. I may need a moment. Quorum sensing is the form of cell-to-cell communication in bacteria in which individual cells coordinate their behavior based on population density. In human terms, the word quorum means minimum number of people required to conduct business. During quorum sensing, bacteria produce and release chemical signaling molecules called autoinducers. When the number of bacteria is low, any autoinducer produced may just diffuse away and nothing much happens. However, as bacteria increase in number, so does the density of autoinducers. The increased number of autoinducers is then more likely to be detected by other bacteria, enabling them to coordinate their behaviors as a group. Such behaviors include motility, expression of virulence factors, biofilm formation, bioluminescence, nitrogen fixation, and sporulation. And that's probably not all. But this is a show about viruses, so why am I telling you about quorum sensing in bacteria? Well, a couple of years ago, yes, this story is a couple of years old, but it is just so cool that I have to retell it. A couple of years ago, Justin Silpa and Bonnie Bassler of Princeton University published this paper in which they showed that a phage named VP882 can monitor the density of autoinducers. Since phages are viruses that infect bacteria and are hence obligate intracellular parasites because they need to be inside their host to replicate, their existence depends on the presence of viable hosts. Having the ability to monitor the number of potential hosts would allow a phage to select the best time for launching an infection, while simultaneously ensuring an excess of new hosts for its offspring. It's kind of like, hello, are there any bacteria around? No, I don't sense too many autoinducers, so that means there aren't that many bacteria. So if I infect a bacterium right now, my offspring won't have enough hosts to infect and will probably go extinct. So I think I will hold off. So if a phage like that doesn't replicate right away, what happens to it in the meantime? VP882 is a temperate phage that infects Vibrio cholerae, the bacterium that causes cholera. Temperate phages can switch between a lysogenic and a lytic replication cycle. In a lysogenic cycle, phages integrate their genome into the host cell genome and become latent. They stay put for a while. In a lytic cycle, they actively replicate, make lots of copies of themselves, and kill the cell. If growth conditions are poor at the time of infection, the phage may enter the lysogenic pathway and exist latently as a stable plasmid or integrated prophage. Once bacterial growth conditions become more favorable, the phage can switch to a lytic program, inducing the production of offspring and lysis of the host cell. For the well-studied phage lambda, the lysis-lysogeny decision depends on the activity or inactivity of a protein known as CI. CI is a transcriptional repressor that inhibits the expression of a phage lytic gene named Q. But hold on to that thought for a second. What is a transcriptional repressor? Remember the central genetic dogma? Transcription is the production of RNA from DNA, from the gene, on the journey to becoming a protein. A transcriptional repressor will bind to that gene that needs to be transcribed so that the proteins that are involved in transcription can't get to the template DNA. So to get back to our story, CI represses the production of Q RNA, so Q will never be expressed as a protein. Because Q is a gene that regulates the lytic cycle, If Q is not expressed, then the phage will not be able to enter the lytic cycle and will likely enter the lysogenic cycle instead. Silpe and Bassler found that the phage VP882 encodes a receptor that binds an autoinducer produced by Vibrio cholerae, and this binding induces a phage gene called Q-tip. How cute. Once activated, Q-tip sequesters and inactivates CI protein, and having lost its repressor, Q is then free to activate the phage lytic pathway. In other words, when there are plenty of autoinducers around, meaning that there are plenty of host bacteria, phage VP882 can sense that by binding the autoinducers, which then ultimately causes the repressor to let go of the DNA and allow that gene to be expressed. That expressed gene then helps the phage enter the lytic cycle and make lots of babies. Silpe and Bassler also found that they could reprogram VP882 to target and selectively induce lysis in various bacterial species other than Vibrio cholerae by using different promoters for Q. A promoter is a sequence of DNA to which proteins bind that initiate transcription of RNA from the DNA template. And these promoters are different in different species. This finding provides immense insight into the possibility of using phages as an alternative to antibiotics because it allows for better specificity and timing of bacterial killing. There are many potential advantages to phage therapy. Presently, the most important advantage is that phages are equally effective against antibiotic-sensitive and antibiotic-resistant bacteria. The specificity of bacterial killing by phage would also lead to reduced impact on commensal bacteria, which are typically obliterated by conventional antibiotics. Commensal bacteria are part of our normal flora, and we kind of need them for everything from digestion to proper immune function. In addition, phages are capable of disrupting bacterial biofilms, which are extensive three-dimensional communities of bacteria and can be a huge problem. The use of phages would also lead to reduced incidence of opportunistic infections and reduced toxic effects. Nevertheless, the authors are careful to note that these studies were done in vitro and do not necessarily predict success in clinical applications. There are studies showing that the host's innate immune system may play a critical role in the efficacy of phage therapy. Still, a very cool study though, and I can't wait to see where this takes us in the context of phage therapy. That's it for today. Thanks again for listening. My name is Trudy Ray. I'll see you next time on Catch This.